Happy Tuesday, Limbrick Bio students. I'm saying this at 9:55 Monday night because it was just pointed out to me that my video had no audio. And I don't know why, because it's all there. I could record it. The microphone was on because I'm double checking after I made that mistake once before. And for some reason, the thing just refused to play anything. I couldn't even get to play for myself. I played it back. The stuff wouldn't even record, or the slide wouldn't even do anything. It wouldn't go follow my animation. So I have no idea what happened. So this could be a real fun video real quick. We're going to just make the best of it. If I start to cry at some point, you'll know I've just lost it after having recorded this video twice. But let's hope it doesn't come to that. So why not, right? Let's dive right in. Taking a look at this from the other day. How are instructions for making protein sent to a ribosome? Hopefully by now you remember that DNA is the instructions for making protein, and that's unfortunately the nucleus, and it has to get to the ribosome outside in the cytoplasm. So DNA makes a small copy that's in the form of RNA, messenger RNA, the mRNA, and that's how it's going to happen. So the DNA kind of opens up, the RNA comes in and just adds little bases at a time and makes a short little copy of the instructions from DNA, and that mRNA, now the messenger RNA, that can go out of the nucleus to the ribosome to actually tell the ribosome what to make. We'll see that today, how that's going to work. Let's see now. This was stuff that was in some of the homework that was due Monday night. If this is your original DNA strand, what's the mRNA going to look like? Most of the bases follow the same kind of base pairing rules we've been seeing. So I'm going to go through and just everywhere I see a C, I'm going to fill in a G because C on DNA always prompts the mRNA to be a G. Fill those letters in. And then anywhere I see a G, I know it's still going to be a C. Whether I'm talking about DNA, whether I'm talking about RNA, doesn't make a difference. Those letters are always fine. The A's and the T's are where it gets a little different. So first of those letters I see over here is a T. A T on DNA still tells the mRNA to be an A. So just as T would be across from A in DNA, same thing with RNA. If there's a T in DNA, the RNA still produces an A in that place. The difference is when you get to the A's, because as you remember, hopefully, RNA does not have T's, it only uses U instead. So anywhere that there's an A in the original DNA template, RNA is going to have a U to be complementary to that base. So I'm going to go now and fill in U's here. And I think most of you that I was seeing little mistakes with this in the last week or so, I was able to email and get in touch with. I think there's a handful of you I still maybe haven't talked to, but I'll probably send an email in the next couple of hours once I double check that stuff, or tomorrow. But otherwise looking pretty good. So I think you guys have pretty much gotten the gist of this stuff, and hopefully don't have too much trouble going forward. So let's get into what we're doing today. So I just mentioned the instructions from DNA being kind of converted into mRNA and going out to the ribosome so it can make protein. But how does that actually happen? As we've seen a lot by now, again, the DNA is the instructions. It's in the nucleus. It's the recipe for making proteins and living things. A main idea of biology, what we call the central dogma of biology, is that one gene codes for one protein. So every instruction only tells how to make one thing. That's sort of common sense, isn't it? So if you call a phone number, you know where you're calling. And if you follow a recipe, you know what you're cooking, in theory at least. So one gene equals one protein. One set of instructions only tells you how to make one thing. So if we want to go into a little more detail with this, we saw the other day how DNA is so big and important that it can't really leave the nucleus, so the small mRNA copy of a gene is made. That's the transcription process. It goes out to deliver the recipe of the ribosome to actually make the protein. The next step in this whole big picture, the process of a ribosome really making the protein by following that mRNA recipe, is now called translation. So there's these two T processes, transcription and translation. Transcription comes first alphabetically, and it is the one that comes before translation also. So if that helps you remember that, hopefully that's good. And that's what we're going to look at today. We're going to draw some pictures of this. It's going to take some time, unfortunately, for me having to do it twice and you having to actually sit and listen to it. But I've given you in your notes a couple of, I think, 5 boxes. And we're just going to kind of go through step by step and see how this whole thing works. So strap in and get ready to do this. So once again, making protein, that word synthesis, meaning to make, is called translation. That's the name of this process we're going to look at today. And here's a little rundown of what we're going to say. So the original instructions from DNA are in a 4-letter code, or the A, T, C, and G language. And this eventually needs to be translated into the language of amino acid and protein pieces so that the ribosome knows what to make. That's what that word translation is going to refer to. The ribosome needs to know what it's going to make, so we'll use a little Rosetta Stone style translation key that we'll see later today or Thursday. I guess it's later today. Like you might use an English-Spanish dictionary or Google Translate to know what it's supposed to put together. It has to look at these A's and T's and C's and G's and U's and know this is the thing I need to add. This is what I'm going to cook, which doesn't really make sense. We can't read that language very effectively. The ribosome needs to know how to do it. So I want you to think of this whole process like someone cooking. DNA is really just a big cookbook. The cell doesn't need the whole book. It only wants one recipe at a time. So when it knows it wants to make the one protein, it just has to go and make a photocopy of that one page, the one recipe. And that's what the mRNA is. So that's that small mRNA copy, and that's the transcription stage when you make a small short copy of one little piece of a recipe from the big giant DNA cookbook. So next that recipe goes out to a ribosome, who's the chef of the cell. The ribosome reads the recipe and will cook up the protein. So to do that, it needs whatever ingredients the recipe calls for. In this case, the amino acids that form the protein. And it takes those amino acids and links them together in a big protein chain during the step called translation that I mentioned a second ago. That's what we're looking at today. We're going to draw this translation and see how this whole thing plays out. It translates the instructions to know what it has to actually do. It's like if you ever had some kind of decoder ring as a kid, or have ever done some kind of puzzle where it's a code and you have to figure out what letters stand for what letters, like that. So hopefully that analogy of the chef makes things a little bit easier and clearer going forward. For now, let's see what we're actually drawing. In your notes, you have several pictures that look a lot like this, numbered 1 to 5. It's not so much that there are actually 5 steps in this process of translation. That's just how I'm going to break it down, and to show you what happens. It's more like 3.5 steps, really, but we'll take a little bit of time. To get a sense of where we are here, notice that we're looking at part of the nucleus, which is surrounded by a membrane or nuclear envelope. So your nucleus, as it says right here, is inside, and there's this membrane around the nucleus right here, just as a cell has a cell membrane around the whole big picture. Inside the nucleus, we see some double-stranded DNA, which looks much too large to fit through the pores in the membrane if it wants to get to the cytoplasm outside. And realize, again, you don't really want it to go outside the nucleus because it could be dangerous. You don't want it to have the chance of getting damaged. Outside the nucleus, in that cytoplasm, we see a ribosome, and at this point, it's really just hanging out until it's needed to make protein. That's our setup. Now we can see what actually takes place here. At some point, the cell might need to make protein. So it'll unwind a small section of the DNA so that one gene of bases is exposed. That's one recipe for one protein. I'm not going to draw that here, but just picture this little double helix unzipping and basically forming 2 little single lines with the letters on each side, as we did a little bit last week. But anybody see a problem here? There's 2 strands of DNA in the molecule, right? So how do we know which one is the recipe we want? It starts with AAA here on one side, the other one says TTT. How do we know which one we're going to read? They're opposite letters. What if we read the recipe backwards? The ribosome might make the wrong thing. We need to know which one we have the recipe on and which one is just kind of the other guy, right? Well, as you can see here, one of them is actually marked. So cells have ways of marking the DNA molecule to distinguish each side. And the one that gets copied is called a template strand. Just as you might have, a template to draw something or build something or some kind of pattern, something like that. The other guy is just called the complementary strand. That's really just the other side. That's not the one that really is important. The one that's marked these little kind of starbursts here, that's the important one. That's the one we're actually going to focus on. So I'm actually going to go through and just kind of highlight that strand right here for a second. That's the one we're going to look at right now. That's the one that's going to be important. And that's the one we want to make sure that we make our copy of. So that's the one that's going to be the template for our messenger RNA. So I said I'm not going to actually show you the DNA strand opened up, but we're going to take that DNA template strand right there and draw what our messenger RNA would look like from that. And that will then be able to leave the nucleus and go to the ribosome. So I'm just going to draw it right on the side here. This is my DNA right here. That's the original. And I'm now going to switch colors and just give myself a little single-stranded messenger RNA. So up at the top, well, let's start at the bottom actually. I see TAC on the DNA. So what letters would that be for mRNA? Well, that 3 letters right there. If it's a T, that'll put an A there. A would make a U, and C will end up looking like a G. I'm going to do the same thing then in the middle. That says CGC, so that would be then a GCG. And at the top, it's 3 T's, so that would be A. So this red structure right here now is my mRNA copy of my original DNA molecule. That single-stranded smaller piece of messenger RNA is now able to leave the nucleus, go out to the ribosome, and carry the instructions with it to make the protein. So you probably don't recognize this thing unless you spend a lot of time in teacher's room, but this is what a Scantron machine looks like. So you see the Scantron papers here, and how this works is you mark them on your test, then we feed through a key, and eventually take your papers and just push them through the machine in this direction here, and it spits them out the other side over here with the answers all marked in and says this kid got a 70, they got a 90, whatever. So when it's working properly, it's a really easy process. Sometimes we have to go back and double check the answers because it doesn't work for some reason. But that's really a lot of how a ribosome works too. So when the ribosome is working properly, the messenger RNA instructions will go in one side, the ribosome reads those instructions, knows what it says, and it then takes in the amino acid pieces and just pushes a completed protein out the other side like a little conveyor belt as it puts them together. So that's what we're going to draw now, and we'll go on with the process that we just started a second ago. I'm going to go ahead and take that messenger RNA molecule we just drew a second ago, the smaller one, and just draw it now being kind of fed through the ribosome as I just showed you with the Scantron machine. So I'm going to start out here. We're in the cytoplasm now. I'm going to kind of go like this. I'm going to start with the first three letters we wrote. We drew them at the bottom before to match up to the TAC on the DNA. So I'm going to fill those in right here now, and that then makes it AUG as my first three letters. And the middle ones over here were GCG. So that would make it CGC. On this way, I just wrote it backwards, but don't worry, it's still CGC. And then the top one up here was TTT. So that would make it AAA for the last little 3 DNA segment there, 3 base segment. So the mRNA copy that left the nucleus enters the ribosome so that the instructions can be read to make the protein. The ribosome, in turn, then reads the letters, or these bases, in the messenger RNA 3 letters at a time. And each 3-letter segment is the instruction for a different amino acid ingredient. This 3-letter segment is called a codon, since it codes for an amino acid. Now, if every 3 letters codes for 1 amino acid, how many amino acids will this messenger RNA tell us to put together? If every 3 letters tells us 1 amino acid, then this would be our first codon. That's 1 amino acid there. This will be our number 2 codon. Not that I necessarily call it that, but that'll be our second codon. And that's a second amino acid. Okay. And up here, this would be our third codon. And that's another third amino acid. So this little segment of messenger RNA would tell us how to make a 3 amino acid protein. That's really not very big. We've got proteins that are thousands and thousands of pieces long. This would be a very, very tiny protein. The first 3 letters, the first codon, tell us what amino acid number 1 is. Letters or bases 4 to 6, we want to think of them that way, tell us what the second amino acid is. And the last 3 letters are what we could call maybe bases 7 to 9, we then code for the third and last amino acid in this. So again, 9 bases, 9 letters, read 3 letters at a time, and that would tell us how to make a 3 amino acid protein chain. Before we start really looking at this slide here, just go ahead and redraw the same messenger RNA from the last picture, since nothing's really happened yet. So I'm going to take my time and do the same thing here. So I'm again going to draw just a little piece of mRNA going down here. It was what, AUG that we were looking at already in the ribosome. That's our first codon. Those are the first 3 letters that are going to be read, the first little 3-letter set. And then the middle one here was GCG. Nope, CGC, sorry. GCG was our original DNA. So it should be CGC on our mRNA. And the last one up here that we'll get to eventually is AAA. Okay, so we've got AUG as our first codon up. That's the first instruction for how to make protein. So at this point, the ribosome has read the first 3 letters, the first codon, and knows which amino acid is needed. Now it's time for the other type of RNA, the tRNA or transfer RNA, to deliver that correct amino acid to the ribosome so it can be used to make the protein. So there are these tRNA molecules just kind of floating around the cytoplasm at all times, waiting until they're needed, and each tRNA type carries a different kind of amino acid piece. There are 20 different varieties of amino acids, so there's 20 different types of tRNA just kind of floating around all the time, again, waiting for their turn where the ribosome says, okay, it's you I need, you're up, bring this thing right here. So I'm going to just draw a couple of tRNA pieces, and they have this very kind of distinct shape that we'll talk about in a second, but they're going to look kind of like this. And they're just kind of floating up here on the cytoplasm. I'm just going to, numbers here, but they would just have like 1, 2, and 3. They would just have little letters up here, not the numbers 1, 2, 3, but they would have little letters to match up with the codon at some point. Now, eventually, the ribosome is going to call out for one of these ingredients, and the correct tRNA now will deliver its amino acid to the ribosome by just actually dropping in and kind of plugging in like some kind of outlet with a plug into that ribosome to sort of pair up with the AUG on that messenger RNA. So this tRNA now arrives at the ribosome, and again, sort of plugs into that ribosome and into the messenger RNA by matching its bases, like we've seen already with the DNA and RNA, where the A's have to match T's or U's, and the C's always match the G's. So if this messenger RNA sequence is AUG, what would the corresponding and complementary tRNA sequence need to be so that they can interact? What would the tRNA up here have to look like in order to match up with an AUG? Remember, we're talking about RNA, so if there's an A, this guy would have to have a U, and U would have an A across from it, and G would have a C across from it. And that's, again, just on the bottom of this little thing right here, this little funny shaped tRNA molecule. So we've been talking about how the 3-letter mRNA sequence is called a codon. And now we see that the complementary 3 letters on the tRNA, which have to match up with that codon, are called an anticodon. That really just means that they're the opposite of the codon. So that's exactly really what's going on here, right? We've got the codon right here. These 3 letters are the codon, and this now is just what we call the anticodon. It just means that the letters on the tRNA have to be the opposite, the complementary ones, to what we've been looking at a little bit so far. So does this tRNA molecule kind of remind anybody of anything? We say that the tRNA molecule has a clover leaf shape. Since it kind of looks like a little shamrock, you might see it more as maybe like the club suit in a deck of cards. But each kind of tRNA delivers a different type of amino acid whenever it's actually needed. This amino acid that I'm deciding to draw here as a square, let's say this guy has a square on it, that would just be then delivered to the ribosome. So the ribosome read the instructions AUG and decided, oh, okay, I know that instruction, that's the recipe for this piece. The ingredient is the square amino acid. So maybe this guy over here has more of a triangle-shaped amino acid piece. And these, just 20 things. You could draw them any way you want. I'm choosing to do it like that. Maybe there's another one that floats up here, and it has a little circular-shaped amino acid. And again, they're just kind of waiting out there to be called to work. And then that amino acid, that little square piece, it's going to now get dropped off at the ribosome. It's the first piece added to the growing chain that'll eventually become the assembled protein. And if we want to get really fancy here, we can refer to this growing chain of proteins as a polypeptide. That's a word we saw a while ago. That really just means a giant, big piece of protein. I'm going to go ahead and draw something of what we had a second ago. Now, we just had a little kind of square piece of amino acid delivered. That's the first little building block of our protein. So I'm just going to quick draw that kind of hanging off the ribosome over here to show that that's still there. Now, after the first codon is read, the first instruction is read, and its corresponding amino acid, that square, has been delivered, the tRNA that delivered it is now useless. It's not needed anymore. So it's going to be released back into the cytoplasm, and it'll go pick up another amino acid and just, again, kind of wait its turn until it's needed again. And the ribosome will now simply repeat the process for the other codons. So I'm just going to draw now that little tRNA just kind of going back up here. That was the, what's it called? The UAC. That's what we had. That guy now just gets released. Now the process will get repeated for everything else. So the way this happens is the ribosome is actually going to just move down the RNA 3 bases at a time and read the next codon in line. So that's what I'm actually going to draw here. That's why I didn't draw the mRNA initially. I'm going to draw my mRNA now moving along. So the AUG that we read initially have now been kind of bumped out of the ribosome. They were already read, and the ribosome is actually going to move now down to the next 3 letters in line, and that was the CGC. And we still have those 3 letters AAA out here that we haven't gotten to. They're next. But the first codon got read, AUG, told us that we needed this tRNA up here to deliver this amino acid, and it did its job. The tRNA got booted out, and now the ribosome, this whole thing here, will actually just slide down to the left and move on to the next codon that says CGC. So now we're going to see what happens there. But the whole process is now just going to get repeated. I'm going to go ahead and just erase these little guys. I'm going to leave that guy up there, but we don't need to see the other tRNA or the other arrow rather, because we already finished that. I want to be able to draw that. So this second codon now tells us which amino acid is next needed, and a now second tRNA will deliver that new amino acid. So again, we'll match up what it's going to be. If the codon, let's write that right here, says CGC, what are the opposite and complementary letters going to be? C and G's follow the same rules, so the tRNA now will have to have a G on it with a C and another G, and that again is going to have this little clover leaf shape, and it'll bring in, let's say this is the circular shaped one. Again, you can draw any shape you want. There's 20 of them. They all have these weird shapes and structures. But it's now going to bring its piece, and this whole thing will now just get kind of added right onto the end of that. It'll get added right to the chain that the square piece started, and then this tRNA will also just float away, get bumped out, and the whole thing will go to the next step again. And this process will repeat for every 3 bases in the mRNA recipe. So each codon is the order for a new amino acid ingredient, and then that gets added to the end of the chain, and the ribosome moves down again to the next codon, and so on, and it just keeps doing it as much as it needs to. Right here, we've only got 3 codons, so it's only going to be 3 little amino acid pieces. But again, like I said before, you could have 10,000 amino acids in chain. They can be either really, really tiny or huge. So it depends just on what your recipe is. Now, we didn't look at this before at the very beginning here, but this whole process of translation has to start somewhere. The ribosome needs to sort of be turned on and told, okay, now you're going to get to making protein right now. Otherwise, it's just kind of sitting there. It needs to get that signal that this is now your job. You need to get to it. So the ribosome, in that same way, then needs to know when to begin and where to begin. And for that reason, the first codon in any kind of chain of mRNA that tells it to start reading and making the protein is going to be called the start codon. It's just what it sounds like. It tells it this is where you're going to start. So it reads the instructions, it starts translating the instructions, it calls in tRNA. That's what's going to happen once it reads a start codon. And only there. You may have a bunch of other DNA pieces, or mRNA letters rather, and the ribosome only actually really starts paying attention, doing its job when it sees that start signal. So it's like if you're ever doing a race and you may have like some kind of false start thing, and people are trying to psych you out, they're trying to not so much in a real race you do, but maybe like in gym class, somebody's like almost saying start and trying to get you to go early, and then when you finally hear the word start, then you actually get to go. It's like that lab we did where the little colors were flashing and you couldn't start right away. So this is how the ribosome knows what to do. So let's just go ahead and redraw our thing. Now we already had the 2 codons go through, so I'm just going to finish the process off now. So most of our mRNA has already gone out here. This is the AUG all the way down there, and then was it CGC we had in the middle here. And now our last 3 letters, our last 3 bases on the mRNA, our third codon is AAA. So we had already, what was it, the square piece, and then we had a circular piece added. And let's now say that this one codes for the third one. This would be now UUU will be the tRNA. And let's say that delivers a triangle-shaped amino acid as our third thing here. So that would then just get added onto this chain. And I'm actually going to draw that right here. I'll just add it right onto the end. So this guy passes it off right to there. And now it's on the end of our little protein chain. And this tRNA now can just float off into the cytoplasm, go back to whatever it was doing, and it'll go right away back to minding its own business. So in the same way that the ribosome has the start codon when it needs to actually begin this process, it also needs to know when to stop. So somewhere in this mRNA is now also what's called a stop codon. And this is what's going to tell the ribosome to stop reading. You're done. We've made our whole protein. This is the thing we were trying to make. You're good to go. So in this way, we didn't really draw it here, but somewhere down here, let's say maybe there was an extra couple of letters here that basically just said stop. Maybe this AAA told it add the triangle piece and then you're done. But there's always going to be some kind of little sequence at the end of the instructions that says, okay, this is the last thing we need to add. This is the last ingredients. That's it. You've done your job. Thank you very much. And it'll tell the ribosome now you're free to release this protein out into the cytoplasm. Now it's good to go to do its job. That was our 5th step. So I said it wasn't really, 5-step process. I just kind of broke it down to 5 things, but that's protein synthesis. That's how proteins actually get made. The mRNA makes a copy of the original DNA. mRNA leaves the nucleus, goes down to the ribosome. The mRNA gets fed through the ribosome. Ribosome reads the mRNA 3 letters at a time, 1 codon at a time, and then just calls in a corresponding tRNA or transfer RNA to have the matching sequence of letters so it can deliver its amino acid, deliver its ingredients, so that the little ribosome chef can put all the recipe pieces together. So to reiterate this, the mRNA instructions or recipe has the codons, which are the 3-letter code that tell which amino acid is needed to make the protein. The tRNA, which then delivers those amino acids, do so by having an opposite and complementary sequence of bases called the anticodon. So you can see right here, here's the mRNA with its codon, and the tRNA just has these 3 letters, the anticodon. It's just the terms we use for that. It's not like it's a structure that says anticodon or codon. It's just what we call the one thing that has to kind of compete with and compare to the other thing. Now, this whole thing here, I think, is one of the tougher things we're going to do this year. If any part of this doesn't make sense, I've been saying it forever, but just please reach out. This can be tricky, so definitely ask any questions that you're coming up with as we do this. But at this point, how can we tell which amino acid is actually needed? You know, the ribosome has to read this codon and say, oh, yeah, UCG, I know what I need there. Let's get the AGC in here. But how does it even know that? You know, how does it know who to get in contact with? How does it know what this protein is going to look like? There's got to be some way to do this. So let's say a piece of mRNA has the sequence AGC UCA GUC. Now I see 9 letters here, so that's going to be 3 different codons. Each codon is going to code for 1 different amino acid. So this would be a 3 amino acid protein. But which amino acid? That's the question, right? We've got 20 different amino acids. Is it threonine, alanine, tyrosine? That's the best one. Which one of these things? 20 different amino acids. So how the heck are we supposed to know which one we need here? Luckily, we don't have to actually cook this thing, but how could we know what the ribosome is going to put together? Well, luckily, like I just said, we don't need to know this. The ribosome needs to know what to do. We just want to be able to analyze it every so often and say, oh, I wonder what the ribosome's cooking up today. So to tell which amino acid a recipe actually calls for, we can just look it up, thankfully. We use something called a universal codon chart, which is just this little table with a list of amino acid codes on it. So that's the good news. Now, imagine how many different combinations there are of 4 letters making 3-letter codes. That's a lot. There's 64 combinations of 4 letters that can go in little codons. But again, luckily, you don't ever need to know any of this stuff. You don't need to remember anything. You're never going to be expected to know any of this stuff. You'll always have this chart if you ever need to look it up, and then you just go and actually do that. Here it is, your universal codon chart. So this is the table that is always provided whenever it's needed, just as I've given it to you here in your notes. If there was a Regents this year, you would have had this on your Regents. They would just give you the chart. You don't have to worry about that now, but at some point we're going to see this again, and it's going to be something we use in the next couple of days just for practice sake. So notice up top I've circled the words messenger RNA codons just to remind you that's the messenger RNA sequence that's important to use this chart. It's the only thing you need. DNA sequence doesn't matter. You know, the tRNA, even though it carries the amino acids, that's not important. It's always the messenger RNA because that's what the ribosome is reading, and that's what's going to actually give the instructions. So that's the easiest mistake to make, and the most common mistake people make when they're using this chart is that they forget which one they're looking at. Because there's a lot of letters, a lot of these 3 little letter sequences, but it's right there on the top of the chart. It says messenger RNA, so hopefully you never forget that. I'll keep pointing out too. That's what we're going to translate now, and let's see how we're actually going to do this. Going back to our example before, I'm going to go ahead and write AUG here because that was our first codon for the example we looked at, right? AUG were our first 3 letters. So let's see how we can actually do this. If you see just on the left side here, it says first base, and it's got some letters. That's just going to be whatever the first letter in the sequence is. And up top it says second base, and it has those letters here. And then we don't really use it this way, but you could look over on the right. It says third base. I'm going to show you a shortcut so you don't even have to worry about that. So let me just erase this little bit ink right here. And we're going to look for our first letter, and that's the A. So the first letter, our first base, we look on the left side where it says the first base, and I see my A right there. That tells me now that I'm looking in this row right here because all of these start with A as the first letter. The next letter, the next base is the U in the second place. So I'm going to look for the second base, and that's right here for U. And that now tells me I'm looking in this column because all the things in this column, these sequences, have U as the second letter. That means where those things correspond is right in this box right here. All 4 of the choices here have A as the first letter, U as the second letter. So what's the third one? Well, I can see that it's a G. So right here, I see AUG as this sequence, and it tells me that the corresponding amino acid for that sequence AUG, that codon, is MET or START. The start means that's what the recipe for the start codon is. So AUG will always be the start codon. That's the only one on this chart that says start. So every time a protein gets made, this is the first piece to get added. This little thing called MET. And again, not that you need to ever know this stuff, but in case you're wondering what that is, I just gave this little list over here. MET means methionine. That's just the first of these little amino acids. Every protein that gets made, that's going to be the first one that gets put down. So methionine is always the first thing. We can use this chart to do anything. We'll see a bunch of examples next, but really any little random sequence here. Let me just pick a couple of random letters. Let's go. Let's say it was GUC as your code. Let me erase my ink. If the mRNA said GUC, we do it the exact same way. My first letter on the left is G, so I'm going to now look in this row. The second letter is a U, or let's make that an A because we had U last time. Let's make it GAC. So A right here, I'm going to look in this column, and where they correspond, where they line up here, that's going to be our thing at the bottom. So all these boxes, or these things in the box right here, have G as the first letter, A as the second letter. We're looking for C, so that's right here, and that tells us that GAC is the code for ASP. And as we now see over here on the right, that's just the amino acid we call aspartate or aspartic acid. So that's how you use your codon chart. And let's go through and see some examples of this stuff. I wonder what I was going to have you do here. In this example, we see an original DNA sequence of TAC, CGA, CCT, TCA. So that's the original recipe from the double-stranded DNA. DNA opens up. This messenger RNA now comes in and makes a copy of it, and its complementary base pairing sequence is AUG. Remember U because there are no T's in the RNA, so the A will create a U. AUG, GCU, GGA, AGU. And again, we know from our chart up here that's the messenger RNA we care about. So this is what we're going to look at here to try to figure out what amino acids we have. We do not care about this DNA at all. I'm going to cross right through that so you don't even get a temptation to use this thing. So let's go up to our chart here and try to figure out which amino acids will be put in this sequence. AUG we just saw, right? That was the first one from before. So I'm not going to bother to go through that. That was right here. Hopefully you remember that. That's methionine. That's the start codon that says, okay, begin this, but it's always methionine first in real life situations. So I'm just going to write MET right there. Let's go to the next one. GCU. Let me erase my ink. GCU, the first letter is G, so I'm going to look on the left side. That says I'm looking in this row. C is the next one right here. I'm looking in this column. And these two things cross over and intersect right here. So I'm looking in that box. The third letter is U, so I see it right there. And luckily, all four of these sequences, all four possibilities of this combination of letters, code for the same amino acid. That's alanine, ALA. So I'm just going to write ALA. So I've got methionine and alanine. Why don't you take a second, pause the video, try to figure out what the next two amino acids are going to be. I'm going to erase my ink there so you can see it clear and figure out what GGA and AGU we're going to code for. First letter G, I'm still in the bottom row. Having the second letter G, that means I'm looking up here now. And where they intersect is all the way at the bottom here. And the last letter is A. Again, all four choices in this box are the same thing. So it's going to be glycine, gly, G-L-Y. That's my third amino acid. And one last time, let me erase my ink. I see AGU as my last sequence. So here's my A, and there's my G. Everything in this row starts with A. Everything in this column has G as the second letter. They intersect right here. And AGU is our first choice right here. And that, it tells us, makes the amino acid, or codes for the amino acid, serine, S-E-R. So this sequence of DNA up top that I crossed out produces this messenger RNA sequence, and that gives the instructions for the ribosome to make those four amino acids, make a little protein chain out of those four things: methionine, alanine, glycine, and serine. There's a chart. I forgot about that. All right, so the labels here tell you that the top little box up top there is the first page of your homework, and the one on the bottom is from the last page of your homework. Let's just do a little bit of an example here. I want to point something out, which is especially on the top part here, it says tryptophan is the amino acid. You don't need to know the amino acids, but that's this thing right here. That's the TRP on the top here, and it only has one little sequence that codes for it. So I'm just going to fill that right in here, TRP. If you're looking for that on the chart and wondering which one, you don't have to actually memorize that at all. So that means that TRP's mRNA sequence is UGG. So I could go fill that in right here for mRNA, UGG. And we can kind of work backwards. If we know the mRNA is UGG, then what did the DNA have to start as? Well, every U would have originally been an A. Every G would have originally been a C. So the DNA here, ACC, produced or led to the mRNA UGG being made. That coded on the chart for tryptophan. We still didn't write in the tRNA, and really the tRNA is just always going to look very, very similar to the DNA, except that it has U's where the DNA may have had a T. So right here, the U on mRNA would be an A for tRNA, and each of the G's just goes back to a C. So in this case, your tRNA looks exactly like your DNA. No big difference there. You can just fill that chart in the same way. Any place you start, just you can go forward or backwards, because it's always just going kind of backwards with the opposite the opposite DNA bases or RNA bases. On the bottom examples here, 17, 18, let's at least do 17. I think I said this in the other video I'll give you later in the week that I'll probably have to record again also. This says, it should say mRNA. On your homework, I filled in where it said mRNA. I know I saw that, so don't worry about that. This is a DNA strand now, and this is the mRNA strand, and we want to see what the amino acids are going to be. So we're going to go back up here, and again, mRNA. So if our mRNA is AUG, by now I just know off the top of my head that that's going to be methionine, because we've now done that twice. But let's go to the second one, CAC. I'm going to find C right here, A right here, and they intersect right there. And CAC tells me it's going to be histidine. So I'm going to write that right in there, HIS. UGU, well, I see a U over up top here and a G all the way at the end. So right here where they cross, that's going to be cysteine as my amino acid. CCU, I see a C right here, C right there, and CCU goes for proline, PRO. UUC, that's going to be my top box, U, and UUC right there, that's PHE, that's phenylalanine. GCU, G right down there, C right there, and where they intersect, GCU, that's going to be alanine. I think we saw that before. A bunch of amino acids. And our last one is GAC, G right there, A up top, they cross over in this box right here, and GAC tells us it's again aspartate or aspartic acid, ASP. So that's our, 7 amino acid protein. Use the chart the same way every time. Hopefully it doesn't give you much trouble, but let me know if it does. That is finally it for now. I really hope I don't record this a third time, because I just have no intention to do that. So let's hope this works again. I got this done in just about an hour. Not that you probably care, but you are done for now. So go do the form underneath this video, and then I've uploaded a few practice pages with it. Some stuff we just saw a second ago. The first one is a diagram that looks kind of like what we drew with the tRNA and everything. Just label everything, say what's in the picture, go back and compare. It's not the exact same picture, but they all look mostly the same. The rest is practice problems like we just did with the chart. We're going from DNA to RNA, mRNA, tRNA, things like that. As I mentioned before, just be really, really careful with each part. If it says you're going from DNA to RNA to mRNA or mRNA to tRNA, just be very careful and specific about what it's giving you and what it's asking you, because it can be really tricky to just get overlooking things and do the completely wrong thing. So as I said before, this stuff that we did today, I think is probably the most difficult part of this chapter by far. One of the tougher things we'll do this year. So really just please, 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 if you have any trouble with this, any questions, reach out to me. I did learn today, I think I mentioned to some of you who came to Extra Help, that I would have done an Extra Help this Friday, but there's going to be some kind of district computer maintenance on Friday, apparently, right when I would have done it. So I'm not going to be able to do it on Friday. Instead, I'm going to do it a day early and do it on Thursday instead. So that's the 23rd. So you'll get this video posted up on Tuesday the 21st and Thursday the 23rd. 2 days later, I will have Extra Help. That's going to be at 11:30. I've been doing it at noon, but I'm going to have it at 11:30 instead. And then Also next Monday, the 27th, back to noon at that point. So the work from this chapter, this week rather, for lessons 3 that you just looked at and 3.5 that I'll give you on Thursday, that will be both due on Monday night, the 27th. Next week, that will be basically midnight, 11:59 again, like it was this week. Again, I'll post it up on Thursday, the second video, and the forms, the work, all that stuff will be due next Monday night. Until then, stay well, look out for each other. I'll talk to you in a few days.